Greetings, students. So the purpose of this screencast is to look a little bit further at Mendel's experiments and tie them directly to the events of meiosis. We're going to introduce his two laws of segregation and independent assortment, and then sort of explain how they work in the context of meiosis. So let's get started. So the first thing we need to figure out is, why is it that when we cross this true breeding purple flowered plant and true breeding white flowered plant, why'd they all become purple? We said last time, well, it's because purple is the dominant allele. But let's dive in a little bit closer by looking at the life cycle here. So it may be worth pausing the video and sort of writing this down. And then after you're finished, check this out. Hey, we're back. Look at that. Magic. Did you like that trick? So this is roughly what you just copied down. Now, hopefully you notice that this bears an awful lot of similarity to another drawing that we did the last time I was with you doing a screencast. And we talked about how if a male adult, female adult, well, a male adult has 46 chromosomes, and then in this process called meiosis, cuts that chromosome number in half, producing these haploid sperm cells. Female does the same thing, producing haploid egg cells. Those then come together to form the zygote. Well, guess what? The same thing is happening in our pea plants. So this true breeding purple flowered plant, we already said was big F, big F. That is a homozygous genotype, and the phenotype is purple flower. And that's crossed with a true breeding white flower, little f, again a homozygous genotype, and the phenotype is white. And then in this process called meiosis, which also happens in pea plants, the true breeding purple flower is going to produce sperm, but the sperm is only going to have half the number of chromosomes, which means it only is going to have one F. And the same with this true breeding white. The egg is only going to have one F. So these sperm cells have the dominant big F allele. These egg cells have the recessive little f allele. And then the sperm and egg meet to form the zygote, which is now a heterozygote because it has one dominant, one recessive allele. And then that grows up to be a purple flower because, it's not very legible, sorry. That's supposed to be big F, little f, because the big F is dominant. So we can kind of explain Mendel's, how that purple flower came from the true breeding purple, true breeding white, and also connect it to this life cycle. So let's go back to the slideshow. And so based on his results, Mendel came up with two laws that can be explained really well based on our understanding of meiosis. The first law is the law of segregation. The law of segregation says that the two alleles are going to separate into different gametes during meiosis. So those two big Fs are going to separate out. The two little f's are going to separate out. Let's go back to the tabletop dot cam and see how that plays out. So let's suppose I have a heterozygous pea plant. They've got a big F allele and a little f allele, and they're on different homologous chromosomes. We said before homologous chromosomes are similar, but not identical. They have the same type of genetic information, but they're not identical. And so here's an example of this, right? The red, oh, that's kind of weird that I did it that way. Should I flip it? I'll leave it. So the red has the purple flower allele, and the purple has the white flower allele. And so what happens is during meiosis, and feel free to reference some diagrams if you don't remember this, but during meiosis what happens is those chromosomes replicate, and we get two copies, and we call these sister chromatids, and again we still call these homologous chromosomes. Now, in metaphase 1 of meiosis, these homologous pairs will line up. They pair up like this. And then in anaphase 1, they're going to separate out into different cells. And so that separation that's taking place right here, big F going that way, little f going that way, that is Mendel's law of segregation. So let's go draw that out. So here we have my attempt to sort of capture key parts of meiosis. We start with the big F and the little f. The chromosomes have already replicated. And then in the law of segregation, the big F goes one way, the little f goes the other way. And so you can't quite see, but shoot, it got cut off. But these have the big F allele, these have the little f. That's at the end of meiosis 1. And then, of course, at the end of meiosis 2, these sister chromatids are going to separate out. And so you're going to get 2 gametes, each with that big F allele. Here, same thing. You're going to get 2 gametes, but this time with the little f allele. And so this is the law of segregation. Big F goes one way, little f goes the other. There's a second, and there's like a prettier diagram that might be worth pausing and studying if you're a little confused by that. There's a second law Mendel came up with, the law of independent assortment. The law of independent assortment says that alleles for different genes are going to separate independently from one another. So again, let's go back to our candy bar analogy to see how that works. So here again we have those same chromosomes as before. This chromosome has the big F allele, this chromosome has the little f, and they are homologous. But remember, most organisms don't just have one pair of chromosomes. Humans, for example, have 46 chromosomes. And so what happens if we introduce other chromosomes? So let's introduce some Milky Ways here. And so, now we've got 2 different types of homologous chromosomes. Well, again, in meiosis, what's going to happen is, first thing is the DNA is going to copy itself. So the one chromosome now has 2 sister chromatids, and that's going to happen with the Milky Ways as well. But then in metaphase 1, they're going to line up down the center of the cell like this. And what you'll notice is if they line up like this, when they separate into different cells, you'll notice that the big F's and the big A's are going to travel together, and the little F's and the little A's are going to travel together. But it turns out that's not the only way this could happen. They could also line up during metaphase 1 of meiosis like this. You ready for it? And if they line up like this, well, now when they separate, the big F is going to be with the little a, and the little f is going to be with the big A. And so what this means is that the F and A alleles separate independently from one another. The big F could be with big A, or the big F could be with little a. And so this is a huge source of genetic variation because in a human cell, you would imagine 23 pairs of these chromosomes all lining up independently. So let's take a look and draw that out. So here's a lovely diagram. I guess this should be big A, big B, little b. So imagine the blue is the big A allele, the red is the little a, the green is the big B allele, the yellow is the little b allele. And it turns out that at metaphase 1, they could line up this way, where big A is kind of with big B, or they could line up this way, where big A is with little b. And so when these chromosomes eventually separate out, you're going to get some combinations where the blue and the green are together, and others where the blue and the yellow are together. Sometimes the red will be with yellow, sometimes the red will be with green. That is the law of independent assortment, and it's a pretty complicated idea. Here's another diagram that shows that. So those are Mendel's laws of segregation and independent assortment and how they tie back to meiosis. The last thing we need to figure out is this second result. So we've kind of explained a lot of this, but we didn't quite get to why did we get a 3-to-1 ratio? And to understand that 3-to-1 ratio, we need to introduce a tool called a Punnett square. And that is what the next screencast is going to discuss.